Hello, my name is Krzysztof Koras, and it is my pleasure to present our work on interpretable deep recommender system model for prediction of kinase inhibitor efficacy across cancer cell lines. In this work, we developed the drug efficacy estimation recommender system, or DEARS for short, in which we focused on achieving the state-of-the-art predictive performance as well as the interpretability of the model. So given some input features regarding the drugs and cell lines, the goal is to predict how well a given drug would work on a particular cancer cell line, which in our case is represented as a single number measurement. For the drug input features, we use their inhibition strengths across the panel of roughly 300 protein kinases. And for the cell lines input features, we use the genes coding for those protein kinases, as well as the putative targets of the analyzed drugs and their biological measurements, mostly the gene expression. DEARS is a neural network model consisting of two autoencoders corresponding to drugs and cell line. The input data is first passed and projected into the lower dimensional hidden space, obtaining the hidden representations for both drugs and cell line. And those hidden representations are then passed into the additional forward neural network, which outputs the final drug efficacy estimate. After the model is trained, we perform the post hoc interpretability analysis. First, we focus on assigning the biological meaning to the hidden dimensions of the cell line space. To this end, we first compute the cell lines hidden representations, and we correlate them with the genome-wide gene expression data available for the cell lines, obtaining the rank list of genes per every hidden dimension. Those rank lists are then passed using the gene set enrichment analysis concerning the biological processes, the gene ontology biological processes terms, obtaining the enrichment scores corresponding to processes in every hidden dimension. Then we utilize the drug sensitivity data for drug D, correlating the hidden dimensions of the cell line space with the true response for a particular drug D. And using the previously derived biological processes, we are able to directly connect the drug efficacy to the biological process in the cell line. We evaluate the predictive performance of our model by calculating the root mean squared error and the correlation between the predicted and true values of drug efficacy in three settings. First one, when we calculate those metrics on all of the data points, namely the cell line drug pairs, and also grouping the results per drug and per cell line. So the per drug and the per cell line results here are the medians taken across the analyzed drugs and cell lines. These results show that the DEARS mostly outperforms or performs similar to other baseline models, in particular outperforming the two other simpler matrix factorization approaches. We have also performed the ablation studies in which we replace the continuous inhibition profiles with drug putative target information, observing the significant drop in the predictive performance. As for the assignment of biological meaning to the hidden dimensions of cell line space, we identify 67 biological processes, which were the most represented by the cell lines hidden dimensions. And importantly, the hidden dimensions are roughly independent regarding the biological processes which they represent. We then show the case studies corresponding to three particular drugs. The first and second row show the feature importances derived using the integrated gradients method for drug features and cell line features. And the third and the fourth row utilize the hidden space of the of cell lines, showing that using only two hidden dimensions, we can identify regions corresponding to better drug efficacy, And also, in some cases, only one hidden dimension is quite indicative of the drug response. And we can list the associated biological processes which are the most important for driving the response for these particular drugs. Lastly, we extend the idea from this figure by computing these correlation coefficients between every hidden dimension and each of the analyzed drugs, obtaining a map directly connecting the drug efficacy to the biological processes in the cell lines for all of the analyzed drugs. With this map, we can identify biological processes which are the most responsible for driving the drug response, which facilitates the better explainability of the model, as well as we can compare different drugs concerning, for example, their target pathway or putative targets. So to summarize, we showed that the 10 hidden dimensions are able to capture important information for drug sensitivity prediction problem. Our tailored interpretability analysis helps to explain the model in terms of higher level biological processes. Importantly, this analysis considers features which are not used for training the model, and the ablation study shows the utility of drugs inhibition profiles and accounting for drugs off targets. With that, I would like to thank my supervisor, Ewa Szczurek, as well as all other co-authors and collaborators from both University of Warsaw and Merck. Thank you very much for listening, and I'll be happy to answer any questions. Thank you.